My name is Komlan Atitey. I'm an employee of the NIH, working as a research fellow in the biostatistics and computational biology branch of the National Institute of Environmental Health Science. Today I will talk about my current research work. It's titled Optimizing Single-Cell Spatiotemporal Delay Variations to Identify Key Features Driving Progression. Data reduction is essential for understanding complex processes with high-dimensional variable interactions. Different data reduction methods applied to the same temporal data give different spatial maps. As a consequence, it complicates downstream analysis, interpretability, and data visualization. As we can see in this figure, we apply 6 different dimensionality reduction methods to the same epithelial to mesenchymal transition dataset. All the 6 methods provided different spatial maps. This highlights the necessity for robust visualization metrics and benchmarking models, particularly when ground truth data is absent and when the data are collected at different times, such as in the case of temporal single-cell data. This example gives the goal for presenting features for good visualization. It shows a single-cell data collected at multiple time points. The data is clustered and projected to a low-dimensional space or reduced space, with the goal to demonstrate the dynamic relationship among cells in the projected space. The high-dimensional data should be clustered based on multiple time points at which the data were collected. To this end, we propose a multi-state Markov-based computational framework denoted as a time-ordered structural learning to tackle the spatiotemporal dependency of ordered cells in the low-dimensional space resulting from temporal data reductions. The framework is composed of 6 main computational sections. For example, as illustrated in this figure, the first step consists of partitioning the reduced data to create several consecutive 2-square submetrics for Markov chain manipulation. Secondly, considering cell states are varying discretely with time, we next estimate the probability of cell being at a chosen state at the next consecutive time step. Our computational framework model time-dependent pattern in data and can handle dataset with any data point ordering. It expands 2-dimensional data into a large matrix to capture how the data change over time. Here, we provide an example of a Markov chain with 5 cells at 5 different states with selected state transitions. The state transition probability matrix of the Markov chain gives the probability of transitioning from one state to another in a single time unit. Thirdly, we compute the expected number of times each individual cell taken from its state could visit other state of cell in the Markov chain. As output, we generated a WW square matrix describing the transition of all the cells in the possible W state corresponding to the number of rows of the reduced data. Fourthly, we weighted each row of the matrix WW square by the proportion of associated level of cell type in the reduced data. Fifthly, we computed the cumulative sum of the weighted WW square matrix. And finally, we calculated the column mean of the WW square matrix and return it in a row vector of length W. We use the mean to estimate the expected variance that describes the amount of variability of data around the expectation. This figure resumes all the steps of our computational framework from the partition of the reduced data to the width of each row of the WW square matrix. Our computational framework uses the entire process, which leads to the stationary distribution of a Markov chain of W total step accounting for the number of rows of the reduced data. It assesses how a trajectory method accounts for the time ordering variations in the spatial structure of the data by investigating how the variance converge to the equilibrium. As proof of concept, we apply our method to evaluate the spatiotemporal trajectory performance of the C-dimensionality reduction method in single-cell analysis. The evaluation was performed on the basis of using 3 different single-cell data, including epithelial mesenchymal transition data, chemical-induced pluripotent stem cell, and spermatogenesis dataset. The results show that more the number of cell types in the data is high, more the delay time for stationarity is long. So the delay time for stationarity depends on the complexity of the data. With the aim to improve our computational framework for feature selection, we related our new project to the understanding mechanism during beta cell development. The goal of this project is to identify key genes that are responsible for the differentiation of the knockout beta cell away from the wild type beta cell. This process is known as causing diabetes. This work motivates our current research in the direction of identifying genes and cells which trigger the stationarity of state transitions of cells during the developmental biology. Also, it motivates our future research toward developing a better data abduction method that preserves global spatiotemporal dependent cellular dynamics for a better visualization. This work is funded by NIH Division of Intramural Research. Thank you for your listening.